So today we're going to talk about DNA replication. DNA replication, which happens during the S phase of the cell cycle. Right? So before we go into DNA replication, we have to understand the structure of a nucleotide. So here I'm going to draw a simple structure of a nucleotide. So we have the nitrogenous base, right, that includes the A, C, T, G. And then we have the pentose sugar, and then we have the phosphate group. A nucleotides, you have to focus on the number of carbons, right? The pentose sugar has 5 carbons. So I'm going to locate the number of carbons. We call this carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 over here. Okay, so a nucleotide, it will only focus on the differences between the pentose sugar. DNA is nucleotide is made of a deoxyribose sugar, while RNA is composed of a ribose sugar. Okay. They both have an OH at carbon 3, but for DNA, it says deoxyribose. For DNA, carbon 2 only has an O. Okay, carbon 2 has an O. No, sorry, carbon 2 only has an H, while for RNA, its ribose is made of OH. Okay. So this is deoxy. Then this is for ribose. Okay. So both DNA and RNA has an OH at the carbon 3. So what it means is that whenever the DNA polymerase or RNA polymerase is adding a new nucleotide, it will always add to the carbon 3 location. New nucleotide is always added to carbon 3. Hence, you have the saying that says that it always reads from 5 to 3. Right? Because the DNA polymerase or RNA polymerase will always move in the 5 to 3 direction. 5 prime, 3 prime direction. Okay. So now, let's get to the double strand of the DNA. I'm going to draw a double strand of DNA. And I'm going to label this as the 5 prime, which represents the 5 prime end of the nucleotide. So that's the phosphate group's end, and we have the 3 prime end over here. Because DNA is anti-parallel, the opposite strand has to be 3 prime to 5 prime. Okay. What I'm drawing here, right here, is the sugar phosphate backbone. Which is made of the sugar, the pentose sugar, as well as the phosphate group. And in between are the bases, right? Because DNA is a complementary base pair, if one side has a nitrogenous A, then the opposite side must have T. A pairs with T, C pairs with G. So I'm just gonna randomly set this sequence from 5 prime to 3 prime at the opposite strand is going to be A, T, G, C, T, A. So the opposite strand has to be complemented, right? So this is going to be T, A, C, G, A, T. And in between those bases, they are connected by hydrogen bond. So A and T, T and A, GC, GC, TA, and AT, right? So in between them are hydrogen bond. This is the attraction that holds the double strands together. So I have labeled the double-stranded DNA. During the synthesis, the process of DNA replication, what will happen is, in my drawing, I will skip and simplify the nitrogenous base sections, and I will only focus on the backbone. So you have to imagine it yourself that the bases are still there. It's just that I kind of simplify it, and I'm not drawing all the bases out. Okay, so during DNA replication, at the original replication, right? So I'm going to redraw the double-stranded DNA. 5 prime, 3 prime, 3 prime, 5 prime. Somewhere in the DNA, right? There's this point over here called origin of replication. Right? This is the place where DNA will start its replication process. So this is location where DNA begins DNA replication begins. Okay. At this time, the double-stranded DNA are still held tightly by the hydrogen bond. A specific protein called helicase is going to bind at the origin of replication. Right? So one helicase at the top, one helicase at the bottom. Right? It's an enzyme, and this enzyme's specific function is to unwind the DNA as it breaks apart the hydrogen bond between the DNA bases. Okay. And they're going to move in the opposite direction. Helicase at the top is going to move in that direction, and this helicase is going to move in this direction. Okay. So helicase is going to move forward in the opposite direction to unwind the DNA. So you can imagine that as the helicase moves in the opposite direction, right, the DNA will start unwinding itself. So I'm going to start drawing the unwinding process. So this is what happens when helicase moves forward. Okay. Oh. All right. And here, the bases are still being held together by the hydrogen bond. As the DNA is unwinding itself, right, it's going to create a situation called a supercoiling situation, right, a supercoiling. Supercoiling is going to create a lot of strain towards the DNA because it's going to imagine it getting a squeeze, like a ropes as it's being opened. At the end part of the ropes is going to entangle and twine with each other even more, and is going to break the ropes, right? It's the same thing for DNA. If you start unwinding the DNA, you're going to create a lot of stress at the end part, right? In front of the helicase. So what happened is you have a specific protein that comes to the rescue. And this protein is called topoisomerase. So topoisomerase is going to come in and is going to relieve some of the break created by this supercoiling situation. So the way how it does it is it will bind and break apart the DNA. It will shrivel and then rejoins the DNA in order to relieve the strain. Caused by unwinding. Okay. So there's another topoisomerase. Okay. So the locations in which the helicase is going to open up, right? This Y-shaped location here, we call it a replication fork. Replication fork is basically the Y-shaped region when the DNA opens up. Right? So this is another replication fork. And when helicase open out, right, this region over here, they are now single-stranded, right? Because they do not form hydrogen bond in between each other, right? They do not form hydrogen bond in between these two strands. So you need to protect this single-stranded DNA from rebinding with each other again. The way how it does it is there's some single-stranded binding DNA that binds at the single-stranded DNA. Okay. So they're just going to temporarily bind it in order to stabilize the DNA. Single-strand binding protein to stabilize the unwinding DNA. It's going to bind there, stabilize it, and when DNA polymerase comes in and starts synthesizing a new strand of DNA, they're going to leave. Okay. So as the helicase moves forward in the opposite directions more and more, the replication bubble is going to become bigger and bigger, right? This region over here is called a replication bubble. So as the helicase moves further and further, the replication bubble gets bigger and bigger. So to initiate the replication process, first we have a specific protein that binds. I'm going to use a different color. That binds to the DNA, and we call it primase. Right? So the primase function is to synthesize a primer. So what is a primer? A primer is an RNA sequence that allows DNA polymerase to bind. So RNA primer. So a primer is a sequence that allows DNA polymerase to bind and initiate DNA replication. Okay. I'm going to move this over to here. Okay. I'm going to label the prime for the DNA strand. So the primer is going to synthesize a primer. It's very short. It's made of RNA. And once it finishes synthesizing the primerase is going to leave. So primerase will detach once it synthesizes the primer, which is very short. Right? So the primer is going to stay. The primer is going to stay there. In the meantime, the helicase is continuing its unwinding process, right? Next, we have DNA polymerase. So DNA polymerase is going to bind at the primer location. And it will synthesize the new DNA, right, by adding new nucleotide to the 3' location. Right? So new nucleotide is going to be added, base pairs with the backbone over here, right? This is what we call the template strain right now, as it will be used as the template to find the correct nucleotide to pair with the bases on the nucleotide of the template. And as new nucleotide is being added, new covalent bond is going to be formed with the primers, and then new nucleotide is going to be added and form a new covalent bond with the previous nucleotide, right? So this will be the new 3', and then a new nucleotide is going to be added, forms a bond as the DNA polymerase moves forward. So DNA polymerase is going to move in these directions. Okay. And as you can see, because this strand is getting synthesized in the same direction as the opening of the DNA. Remember that this is the replication fork. Because helicase is going to open out the DNA in the same direction as how the newly synthesized DNA elongates, right? They are facing towards the same direction. It means that this DNA polymerase will just keep moving and moving and moving. So we call this newly synthesized strand a leading strand. So this is a leading strand. So what is a leading strand, right? A leading strand is a new DNA strand that is synthesized continuously with non-stop. Okay. So same thing over here in the opposite strand, right? The primase is going to synthesize a RNA primer 5' to 3'. As the primase moves forward, it will detach. And then DNA polymerase, my bad. DNA polymerase is going to bind here. And it will start moving in the opposite directions from 5' to 3'. Right? So newly synthesized DNA is going to elongate in this direction. So this is also the leading strand. Because this DNA polymerase is going to move to the same direction as this helicase over here. Right? Here's the replication fork. Okay? So what happens to these strands over here, right? Because this DNA polymerase, right, is unable to move in the 5' directions. It just can't. So it has to move to the right, which is the 3' directions, as new nucleotides is being added at the 3' over here. So what happened is a new primer has to be synthesized here with the help of primase. So primase comes in, synthesize a new primer. Then the primase is going to leave. And then another DNA polymerase is going to come in. And it will start moving in the 3' directions. Right? As you can see, as this DNA polymerase is moving to the right, which is the 3' directions, the back over here, right, is where the helicase is located, right? This is the helicase and is going to move in this directions to the left, which is in the completely opposite direction as where DNA polymerase is heading to. So we call this strand a lagging strand, right? So this is a lagging strand. Because this DNA polymerase will move until it reaches the end, right? Or the end of this newly synthesized DNA as it encounters the primer in front of it. So this DNA polymerase will move, right? It will move and then it will encounter the RNA primers in front of it. And it will detach, right? So this DNA polymerase will eventually detach once it's finished synthesizing. Once this reaches the primer. Okay. So let's focus here, right? Now this DNA polymerase has, start synthesizing, it's moving forward, it is a leading strand, primers has gone, has left already. Okay, and now we have the lagging strand synthesizing a new strand of DNA, and it reached the primers. DNA polymerase detach, and now we have to replace this primer over here because the primer is made of RNA. Right? This primer is made of RNA. So another DNA polymerase is going to come in. Right? Another DNA polymerase is going to come in. And what it does is it's going to replace the RNA primer with DNA nucleotides. Okay. And once it finishes replacing all the RNA primers into DNA, it's going to detach. And now it has been replaced with DNA. But here, there's still a gap between the lagging strand and the newly synthesized DNA after you replace the RNA, right? There is a gap in between here. So what happened is a ligase is going to come in . A ligase is going to come in, and it will form a covalent bond. It will form a covalent bond between the nucleotides. Okay. And I forgot to mention the keywords, a keyword over here called Okazaki Fragment. So Okazaki fragments are the pieces of DNA that is synthesized in the lagging strand. So this is an Okazaki fragment. I don't have enough space to write. Okazaki fragment. And the same thing happens here, right? The new primer is going to be synthesized, and the lagging strand is formed as the DNA polymerase binds at the primers and it moves in that direction, synthesizing an Okazaki fragment. Lagging strand. So with this, I have finished the replication process. Okay. Some of the keywords that you need to be aware of is DNA polymerase, right? Ligase, different type of DNA polymerase. The number of DNA polymerase is not that important. Leading strand, lagging strand. Primase, Okazaki fragment, DNA polymerase, replication fork, and the directions of how to add new nucleotides into the newly synthesized strand. Peace.